let's talk about our old friend Legionella. But first, let's talk about copper. Copper is an antimicrobial metal, which is fancy science talk for saying that it's able to kill bacteria. Now, copper has been used for millennia worldwide in medicine. Everyone was using copper. From ancient Babylon to ancient China to pre-Columbian North America, everyone was using it. In ancient Egypt, they used copper drinking vessels to help sterilize their water. And this papyrus, the Edwin Smith Papyrus from 1500 BCE, actually details the use of copper solutions to treat wounds. The Greeks used copper in much the same fashion. Hippocrates, often considered the father of Western medicine, described using copper solutions and powdered copper in treating wounds, infections, and eye injuries. But while they were using copper, they didn't know how it worked. We've only had the tools to understand how copper can kill bacteria for a pretty short period of time. And even in that time, the focus wasn't on antimicrobial metals, it was on antibiotics. So we don't know a lot about how copper kills bacteria. We know a couple of things. We know it is extremely complicated and that it has multiple pathways. Arguably, the main mechanism copper uses to kill bacteria is through causing protein misfolding. Now, what that means is that copper ions are preventing the formation of functional proteins and functional enzymes, and this disrupts the metabolism of the cell and leads to cell death. Copper ions poke holes in the cell membrane, allowing them to go into the cell and the cell contents to leak out. Copper ions can also cause damage to the cell wall, weakening the cell. And finally, copper ions react with oxygen to form toxic oxygen radicals. These radicals are highly reactive, and they do pretty much what the picture showed. They blow up the DNA, and this DNA damage leads to the death of the cell. Unfortunately for Legionella, it encounters copper in a couple of different places. So as a quick refresher, I know it was pretty recent, but Legionella causes Legionnaires disease. It's found in the natural environment, so in lakes, ponds, and rivers, but it's mainly found in man-made water systems, like air conditioning systems, cooling towers, showers, hot tubs. And within these systems, Legionella infects other microbes, like amoebas. And from these water systems, Legionella is aerosolized, or sprayed to the air in droplets. And we can breathe in these droplets. Once inside of our lungs, Legionella invades our immune cells. And it starts to replicate inside of our immune cells just as it would inside of an amoeba. And this is what causes the infection. So like I said, it encounters copper in a couple of different places. The first is through treatment. Copper-silver ionization is a mechanism that we use to kill Legionella in water systems. So it works by installing two anodes, one of copper, one of silver, and then electrifying them to release copper ions into the water system. And we use this to kill off Legionella. But there's a problem. Because in a lot of water systems, especially in Montreal, where there's a lot of old water systems, there's copper pipes. And it's thought that these copper pipes act as a selective pressure on the bacteria in the water, selecting for copper-resistant Legionella. Now, when we talk about emerging resistance, normally we talk about the acquisition of new resistance genes, whole chunks of DNA, whole new genes, hundreds of base pairs, thousands of base pairs. But in a 2021 study, it was found that from within the same water system, copper-resistant strains of Legionella and copper-sensitive strains of Legionella had only 29 differences in their genetic code, meaning that copper resistance can emerge through just a few small changes to the DNA of Legionella. So bacteria are really, really good at evolving and adapting to new environments. They're taking up DNA from their environment, and because of how fast they replicate, even rare mutations happen all the time. So if we take a population like this, and in yellow we have sensitive bacteria, and in red we have resistant bacteria. We have a population, and it's mostly sensitive. It's a pretty standard bacteria population. And we apply a selective pressure, maybe copper. Now, this selective pressure kills off most of the bacteria, but the ones that survive are the ones that contain mutations that are favorable and allow them to survive the selective pressure. And these resistant bacteria go on to form a new population, one that is resistant to that selective pressure. Copper is no different. It's thought that poor usage of copper, so not using high enough concentrations in copper-silver ionization, or having copper pipes in your water system, will just select for resistant Legionella. Now hopefully I've made it clear that this is stuff that we've thought about. We think about this stuff, we hypothesize. No one's actually demonstrated it. So that's where I come in. I hypothesized that exposure to copper selects for favorable point mutations that help Legionella and pneumophila survive in environments with copper. And I'm doing this with a technique called experimental evolution. So I took Legionella, I grew it up at 37 degrees, I washed and resuspended in water, and exposed that to copper. Then I grew whatever survived, washed and resuspended in water, exposed to copper ions, grew what survived. You get the picture. I repeated this 40 times, so 40 generations. And then I took my evolved strains, and I extracted the DNA and sent it off to the Genome Center for sequencing. Once I get those sequences, I will analyze the genomes to identify any of the mutations that conferred copper resistance. And then I will induce those mutations in the ancestor of these evolved strains to examine how they affect copper resistance. Before I can show you my results, I do have to go into a bit more detail though. So I had the ancestor strain, Philadelphia 1. This is the one that was found in 1976. I streaked out on an agar plate, and I took 12 colonies. 6 of those colonies I diluted in water for 40 generations. These are my control lineages. The other 6 are the ones that I repeatedly exposed to copper over the course of 40 generations. These are my evolved lineages. Okay, I have my control lineages, I have my evolved lineages. Now I need to make sure my evolved lineages are resistant to copper. So I took my control lineages, and I diluted them with water for 4 hours. These are the untreated control lineages. I took those same control lineages, and I exposed them to copper ions for just 4 hours. Those are my treated control lineages. Then I took my evolved strains, or evolved lineages, and I exposed them to copper ions for 4 hours to form the treated evolved lineages. Once this exposure was done, I plated them all on agar plates, and I counted the number of colonies. And there were a lot of colonies, so I put them on a graph. On this graph, I'll show cell survival. On the y-axis, we have the number of cells, so I use a log scale, and that roughly corresponds to a certain percentage of survival. So 1 log reduction is 1% survival, 1 log is 10%, 2 log is 1%, so on and so forth. Okay, so I took my control strains, I exposed them to water, untreated, 100% survival. There's nothing in there that could kill my Legionella. I take those same strains, I expose them to copper ions. There's an average of a 2 log reduction, which again, fancy science talk for 1% survival. These are not copper resistant at all. It's a pretty significant reduction. But I'm going to be honest, we're not here for the control strains. We're here for my evolved strains. Are they actually copper resistant? Well, when I exposed them to copper, they survived just as well in the presence of copper as my control strains did in just water. Meaning, I actually did in fact make copper-resistant Legionella, which is very exciting for just me, because it means I can continue my project. So like I said, I have extracted their genomes, I've sent them off to the Genome Center, and I am eagerly anticipating receiving those results back, so I can start to compare the genomes of my evolved strains and identify those mutations conferring copper resistance. Once I've identified those mutations, I will do exactly what the lady in the picture is doing. I will take the individual base pairs and I will change them. So I'll take Philadelphia 1, the ancestor, and I will create a series of mutants, each containing just one of the point mutations possibly conferring copper resistance. Then I'll take those mutants, I'll expose them to copper, and I'll see how the mutations affect a survival in the presence of copper. Okay, to summarize. Copper is an antimicrobial metal, and we use it to kill Legionella. But there's a problem because copper resistance is emerging, and it can emerge pretty quickly through just a small number of changes. It took me 8 months for those 40 generations, which is not very long on the evolutionary timescale. And poorly using copper, so not enough copper ions to kill the cells, or having a copper pipe just creates resistant Legionella. And we can take this knowledge so we can more effectively use copper in Legionella control. And that way we can better protect people from getting Legionnaires disease. So that's all I have for you today. Thank you so much for listening.